Hello, everyone. A quick note before today's story. This one is on the complex side, but incredibly important. So much so that there are bits and pieces of today's topic scattered throughout previous episodes we've done. We've tried our very best to take this complex topic and make it easier for everyone to understand. With that said, fire up that curiosity and get ready for today's story.
Hey, you know how sometimes on Saltwater Stories we've hinted at these almost magical ways scientists can tell what's going on in the ocean without even seeing the creatures themselves?
We have, yeah. We've touched on analyzing water samples, finding those little traces of who might be swimming by.
Well, today we're really exploring that whole idea, looking at a truly groundbreaking method for uncovering the ocean's secrets, using what you could call
Well,
invisible clues.
that's right. Forget the nets and the cameras for a moment. Well, not entirely, but this is different. It's about understanding marine life in a completely revolutionary way, deciphering the ocean's mysteries through the genetic material organisms leave behind.
Okay, invisible clues, genetic material. Let's get specific. What exactly are we talking about here?
We're talking about environmental DNA, or eDNA for short. Basically, it's DNA collected from environmental samples like, seawater or sediment.
So not directly from the organism itself.
Exactly. Not from a tissue sample of a fish or a whale you've caught or tagged. It's the DNA shed into the environment. Think of it as the collective genetic signature of everything that's been present in that particular spot of water or patch of mud.
Huh. So if you just scoop up some seawater, you might actually be holding tiny fragments of DNA from everything that swam through recently. Sounds a bit like forensic science.
It's a great analogy, actually. It really is similar to how forensic scientists analyze a crime scene. They look for fingerprints, maybe hair follicles, fibers.
Got its stuff left behind?
Exactly. Marine scientists using eDNA are looking for those genetic traces in the water or on the seabed. Genetic fingerprints, if you will.
That makes a lot of sense.
And-
Okay, so where does all this DNA floating around in the ocean actually come from? It's not like sharks are just, shedding their entire genome constantly.
No, not quite. But organisms are constantly releasing bits of themselves. Think about us. We shed skin cells all the time. Marine organisms do too. Fish shed skin cells and scales. Marine mammals lose hairs. Those are major sources of eDNA.
Like my dog leaving fur absolutely everywhere.
Yeah.
The ocean's just a much bigger couch.
Precisely. Then you've got secretions, things like mucus. You know how slimy some fish are?
Yep.
And excretions, feces, and urine. Terrestrially, think guano from seabird colonies. That's a huge source washing into coastal waters.
Okay, so skin, scales, mucus, waste products.
What else? A big one is gametes. When fish or invertebrates spawn, releasing sperm and eggs into the water column, that's a massive release of DNA.
Wow, I hadn't thought of that.
And of course, predation events or natural death. When an animal gets eaten or when it dies and starts to decompose, its tissues break down, releasing DNA fragments, eventually its whole genetic code, into the environment.
So it's this constant cycle of shedding, excreting, reproducing, and decomposing that creates this pool of genetic information. Anything else?
Yeah, a few other things. Saliva, maybe blood from injuries. And don't forget material washing in from land plant bits, pollen. Plus, a huge component, especially in terms of sheer quantity, is microorganisms.
Bacteria and algae and things.
Exactly. Bacteria, archaea, fungi, protists, their whole cells are floating around, and when they die and break open, that's called lysis, their DNA spills out too. So an eDNA sample really captures a broad snapshot.
It sounds like a constantly changing genetic soup.
Mm.
But if all this DNA is just floating around, how long does it actually last? Doesn't it just degrade and disappear pretty quickly?
It absolutely does break down. eDNA isn't permanent ink. There are several factors working against it.
Like what?
Well, UV radiation from sunlight is a big one, especially near the surface. Water temperature plays a role, so does the pH or acidity and oxygen levels. And then there are the microbes themselves. Many bacteria and other microorganisms actually consume DNA as food.
Okay, so it's under constant attack, chemically and biologically.
Huh.
How long are we talking typically for it to persist in the ocean?
In most typical ocean water conditions, we're talking about fragments lasting from maybe a few days up to perhaps several weeks. It varies quite a bit depending on those factors we just mentioned.
Days to weeks. That's actually not very long, but I guess that's useful in itself.
It's a huge advantage. Because it's relatively short-lived, if we detect the DNA of, say, a specific whale species in a water sample, it's a really strong indicator that that whale was in the area very recently.
Ah, so it gives you a kind of near real-time snapshot of who's currently or was just in the neighborhood.
Precisely. It's fantastic for monitoring dynamic events. Think about tracking a salmon spawning run up a river estuary or detecting the arrival of an invasive crab species in a new bay almost as soon as it happens.
That's incredibly powerful. Does it last longer anywhere else? You mentioned sediment earlier.
Yes, that's an important distinction. Down in the seabed sediments, conditions are often different. It's darker, often colder, maybe less oxygen, fewer degrading microbes depending on the depth. So DNA trapped in those sediments can persist for much, much longer.
How much longer?
Potentially hundreds, maybe even thousands of years in the right conditions, buried layer by layer. So sediment eDNA can give us a historical record, like looking back in time at what communities lived there long ago.
Wow, an underwater genetic archive.
Mm.
So boiling it down, the fundamental idea is you can detect life, figure out what species are present without having to physically see or capture the organism, just by analyzing the genetic traces they leave behind.
That's the core concept, exactly. You take your sample water or sediment, you extract the DNA, and then you use lab techniques to read those genetic signatures and identify the species. It's like conducting a census without having to knock on any doors, just by analyzing the mail left in the mailbox, so to speak.
And I can immediately see how this would be revolutionary for studying those really tricky species.
Oh, absolutely. Think about species that are naturally rare or very elusive and good at hiding. eDNA dramatically increases our chances of just knowing they're there.
Or things that are hard to even get near.
Definitely. Cryptic species that blend in perfectly, or animals that are incredibly difficult or dangerous to capture or even just handle. Think about like fragile jellyfish or huge shy whales or deep sea fish that live kilometers down.
Right, you're not exactly going to scuba dive down to tag a viper fish easily.
Not easily, no. And it's also invaluable for the microscopic world, bacteria, phytoplankton stuff you simply can't identify visually in the field. And critically, for detecting organisms at very low density.
Low densities. Like what?
Like the very first few individuals of an invasive species arriving in a new area when you might still have a chance to manage them. Or, on the flip side, finding the last remaining individuals of a critically endangered population. eDNA can sometimes pick them up when traditional surveys might miss them entirely.
So it's not just more convenient, it's opening up possibilities for detection that just didn't exist before. And you touched on this, but it's non-invasive, right? You're just taking water or mud.
That's a massive benefit, especially for conservation. You're collecting environmental samples, not harassing or stressing the animals themselves. For sensitive or endangered species, minimizing disturbance is absolutely critical.
So you get vital information on their presence, their distribution, maybe even seasonal patterns, all with minimal impact. It really is like unlocking secrets with these tiny clues. Okay, so if we want to find these clues, these genetic fingerprints, how do scientists actually go about collecting the samples out there in the big wide ocean?
Right, collection is the crucial first step. You need to get that water or sediment sample that hopefully contains the DNA you're interested in, and crucially, you have to do it without contaminating it.
Contamination must be a huge issue.
It's a constant concern, we'll come back to that. But first, the where and how. The main sample types, as we said, are seawater and sediment.
And I guess how you collect it depends heavily on where you are, coastal versus deep sea.
Exactly. Methods vary hugely. If you're in shallow coastal water, you might wade in, use a small boat, maybe even just a sterile bottle on a pole. For the open ocean, you're typically working from larger research vessels. And the deep sea, well, that requires some pretty specialized gear.
Okay, let's hear about the gear. I'm picturing robotic arms and high-tech bottles.
You're not far off. A workhorse for collecting water at specific depths is the Niskin bottle. It's basically a tube that's open at both ends. You lower it down to the depth you want,
Okay.
and then you send a signal or drop a weight called a messenger, which triggers rubber caps to snap shut at both ends, trapping the water from that exact depth.
Clever.
Very often, these Niskin bottles are mounted on a large metal frame called a rosette, maybe 12 or 24 bottles together. And usually, that rosette also holds a CTD instrument package, conductivity, temperature, and depth. It measures those parameters continuously as the rosette is lowered and raised. So for each water sample you collect with a Niskin bottle, you get simultaneous data on the water's salinity, temperature, and the precise depth. That environmental context is vital for interpreting the eDNA results later.
Makes sense. What about sampling right near the seabed? or in complex environments like coral reefs or hydrothermal vents.
That's where ROV's remotely operated vehicles are invaluable. These are tethered underwater robots, driven from the ship, equipped with cameras, lights, robotic arms.
The ones you see in deep-sea documentaries.
Exactly. They can maneuver very precisely and carry samplers, including Niskin bottles or specialized suction samplers, right to the target spot, maybe centimeters away from a specific vent or coral head. You can see exactly what you're sampling.
Incredible precision. What if you need to cover a large area or monitor a spot over time without a ship staying there for weeks?
Then you look towards AUV's autonomous underwater vehicles. These are untethered, pre-programmed robotic submarines. They can swim predefined routes, surveying large areas.
Like underwater drones?
Pretty much. And some AUVs are now being equipped with in-situ eDNA samplers. They can actually pump water through filters, preserve the filters on board, all while cruising along underwater. We sometimes call them sipping samplers.
Wow. Automation in action.
Definitely. And for long-term monitoring at fixed points, scientists deploy autonomous samplers. These might be moored in the water column or placed on the seabed, and they're programmed to automatically collect and preserve water samples at set intervals, maybe every day or every week for months.
So you can track changes over time. What about more? Low-tech options from a ship.
Oh yeah, absolutely. Often researchers use shipboard pumps connected to hoses that are lowered to depth. The water is pumped up on deck and immediately passed through a filtration system. You can filter larger volumes this way.
Filtering, that seems key.
It is. For surface waters, sometimes just using a clean bucket or sterile bottle is enough to get the water, which you then filter. And for sediments, the tool is usually a corer, basically a tube you push and drop into the seabed to collect a cylinder of mud or sand.
So a whole arsenal of tools. How do you decide on the sampling strategy? Like where to sample? How many samples?
That's entirely driven by the research question. Are you looking for a specific rare species? Then you might target areas where it's likely to be. Are you mapping biodiversity across a region? Then you might sample along transect straight lines across the area or set up a grid.
Multiple depths too, I assume.
Often, yes. Sampling at different depths is common because communities can change significantly with depth. Time series sampling going back to the same spot repeatedly is crucial for understanding seasonal changes or the impact of duns.
And you mentioned replicas before.
Critically important. Taking multiple samples at roughly the same spot, maybe just meters apart, helps ensure your results are robust and not just a fluke. It increases confidence. And the volume of water filtered matters too, anywhere from less than a liter to 10 liters or even more, depending on how much DNA you expect to find.
Okay, back to contamination. With all this sensitive DNA floating around, how do you avoid accidentally adding your own DNA or DNA from the boat into the sample?
Extreme vigilance. It starts with meticulous cleaning of all equipment bottles, filter holders, tubing, often involving bleach washes, followed by rinsing with DNA-free water. Researchers wear disposable gloves, changing them frequently.
Makes sense.
You try to avoid sampling near wastewater outlets from the ship. On deck, you process samples away from high-traffic areas. And crucially, you run negative controls.
Makes that.
You basically process a sample of known DNA-free water using the exact same equipment and procedures as your real samples. If DNA shows up in your negative control later in the lab, you know you had a contamination problem somewhere in your collection or handling process.
Right, a quality check. Okay, so you've carefully collected your water or sediment, maybe filtered it right away. What's the very next step to stop that DNA degrading before it gets to the lab?
Preservation. Absolutely vital. As soon as that sample is out of its natural environment or even on the filter, enzymes present in the sample will start chewing up the DNA. You need to stop that process fast.
How do you do that? Freezing.
Freezing is a very common method, yes. Once you've filtered the water and captured the eDNA on the filter membrane, you can immediately pop that filter into a sterile tube or bag and freeze it, usually at negative 20 degrees Celsius, or even better, negative 80 in an ultra-low freezer if available. Freezing effectively halts enzymatic activity.
But what if you're out in the field, miles from a freezer, on a small boat, or in a remote location?
That's where chemical preservation buffers are essential. These are liquids you add directly to the filter or you submerge the filter in them. They contain chemicals that inactivate those DNA-degrading enzymes and stabilize the DNA structure.
What kind of chemicals?
High concentration ethanol, like 95% or absolute ethanol is a common one. There are also various commercial buffers specifically designed for eDNA preservation, like Coggan's ATL buffer, or products like DNR and a Shield. Some are even non-toxic, like Longmire's solution, which is great for situations like citizen science projects where handling hazardous chemicals isn't ideal. Though some buffers might require specific adjustments to the DNA extraction protocol later.
So you choose the buffer based on the situation and maybe your downstream plans. Ethanol, commercial buffer. Either way, it lets you store samples without freezing.
Exactly. Often they allow storage at room temperature, or at least in a standard refrigerator, for extended periods. It makes fieldwork logistics much, much easier.
Neither storage tips.
Yes, keep the filters out of direct sunlight. UV radiation is bad for DNA, even after preservation. And perhaps the most crucial non-chemical step is meticulous record keeping.
Metadata. Right.
Absolutely. For every single filter, you need to record date, time, precise GPS location, water depth, temperature, salinity if measured, the exact volume of water filtered, the type and pore size of the filter used, and the preservation method. All that metadata is non-negotiable for interpreting the results properly.
Okay, so the samples are collected, preserved, meticulously labeled, and shipped back.
Yeah.
Now they arrive at the lab. What happens next to actually get that DNA ready for reading it?
That's the DNA extraction phase. The goal here is to get the eDNA off the filter, separate it from everything else in the sample, and purify it.
Separate it from the filter material itself.
Oh,
And other gunk.
exactly. You want pure DNA, free from filter debris, cellular debris like proteins and lipids, and especially free from environmental contaminants that could mess up the next steps. We call those inhibitors.
Inhibitors. What kind of things inhibit the process?
Oh, various substances commonly found in environmental samples. Humic acids, for example, which come from decaying plant matter and are often abundant in rivers or coastal areas with runoff, can strongly inhibit PCR. Certain compounds in sediments, metals, polysaccharides, lots of potential culprits.
So the extraction has to get rid of those too. How does it work? What are the stages?
Typically, there are two main stages. First is lysis, breaking open any intact cells captured on the filter to release the DNA inside.
How do you break them open?
Usually with a combination of chemical treatments like detergents that disrupt cell membranes and often enzymes. Proteinase K is a common enzyme used here. It chews up proteins, including enzymes that might degrade DNA, and helps break down cellular structures. Sometimes physical methods like bead beating are used too, especially for tough microbial cells.
Okay, cells broken open, DNA released. What's stage 2?
Purification. This is where you separate the DNA molecules from all that other stuff. The proteins, lipids, salts, filter fragments, and those critical inhibitors.
And how do you purify it? Are there standard methods?
There are several common approaches. By far the most widely used today are commercial extraction kits. Companies like Keegan, Mobio, now Keegan, Mascher-Nagel, they all make kits optimized for different sample types like water or soil sediment.
What makes these kits convenient?
They usually provide pre-made reagents and a standardized protocol. Many rely on silica chemistry. Under specific salt and pH conditions, DNA binds tightly to silica membranes in small spin columns or to silica-coated magnetic beads.
So the DNA sticks and everything else washes away.
Pretty much. You add your lysed sample, the DNA binds, you wash away the contaminants and inhibitors with specific wash buffers, and then you change the chemical conditions, usually with a low salt buffer or water to release or elute the purified DNA from the silica.
Sounds neat and tidy. Are there other ways, more traditional methods?
Yes. The classic method is phenol chloroform extraction. It uses organic solvents, phenol and chloroform, to separate nucleic acids from proteins and other cellular components. It partitions the components into different layers in a tube.
Is that still used much?
It is, sometimes. It can potentially yield more DNA, especially from tricky samples. But it's much more manual. It involves hazardous chemicals requiring a fume hood. And it might not always be as effective at removing certain PCR inhibitors compared to some modern kits. There are also other methods based on precipitation, but kits are generally the standard now for eDNA work due to their efficiency and inhibitor removal capabilities.
So does the extraction method you choose actually affect the final results? Like, could you get a different list of species depending on the kit you use?
That's a really important point. Yes, it absolutely can. Different methods have different efficiencies for recovering DNA, especially fragmented DNA. They vary in how well they remove different types of inhibitors. And there's evidence that some methods might be biased towards recovering DNA from certain groups of organisms over others.
Wow. So the extraction step itself could skew the picture of biodiversity.
It can introduce bias, yes. It's a very active area of research, comparing kits, optimizing protocols, working towards standardization. The idea of a fit-for-purpose protocol is key. The best method often depends on your specific sample type, marine water, freshwater sediment, soil, and what you ultimately want to analyze.
Okay, so we've navigated the tricky extraction process. We now have this little tube of purified eDNA. It's finally isolated. How on earth do scientists actually read that DNA to figure out who it came from?
Right, this is where we get into the analysis.
Yeah.
And the absolute foundation for almost all eDNA analysis is a technique called polymerase chain reaction, or PCR.
PCR, I've heard of that. It's like a molecular photocopier, right?
That's a perfect analogy. It allows us to take the tiny, tiny amounts of specific DNA sequences present in our eDNA extract and make millions or even billions of copies of them. Amplification is key because the original amount of DNA from any single species might be incredibly low.
So how does it know which bits of DNA to copy? It's not just copying everything randomly, is it?
No, it's highly specific. We use short, custom-designed pieces of synthetic DNA called primers. These primers are engineered to bind to very specific known sequences that flank a target region, a marker gene within the DNA.
A marker's gene.
Yes, a specific gene or part of a gene that we know is useful for identifying species. Then, using an enzyme called Taq polymerase, a heat-stable enzyme originally found in microbes living in hot springs, and repeated cycles of heating and cooling in a machine called a thermocycler, the process selectively copies only the DNA segment lying between the two primer binding sites.
Okay, primers target the region, Taq polymerase copies it, repeat many times. Why do we often hear about mitochondrial DNA being used?
Good question. For animal studies, mitochondrial DNA, mtDNA, is a very common target. The main reason is that each animal cell has hundreds or thousands of mitochondria, each with its own small circular genome. Compare that to the nucleus, which usually only has 2 copies of each chromosome.
So there's just much more mtDNA per cell to start with. Increases your chances of detecting it.
Exactly. Much higher copy number means better sensitivity, especially when the DNA is degraded. And speaking of degradation, eDNA in the environment is often broken into small pieces. So the primers we design usually target relatively short fragments of these marker genes, maybe just 100 to 300 base pairs long, because shorter fragments are more likely to have survived intact.
Makes sense. So PCR makes loads of copies of these specific short marker regions. What do you do with those copies to find out what species are there?
Well, there are a few main analytical paths you can take, building on that PCR foundation. One approach is focused on detecting just a single, specific species of interest.
Like, are whale sharks present in this marine park?
Precisely. For that, you can use standard PCR with primers designed only for whale shark DNA. After running the PCR, you visualize the result using gel electrophoresis. You load the PCR product into a gel matrix, apply an electric current, and DNA fragments move through the gel based on size. If you see a band of the expected size for your whale shark target sequence, it indicates presence. If not absence, it's mostly a yes-no answer.
Okay, presence absence. What if you want to know how much whale shark DNA is there? Maybe get an idea of relative abundance.
For that, we turn to quantitative PCR, usually called qPCR or sometimes real-time PCR. It also uses species-specific primers, but it includes fluorescent dyes or probes that glow when new DNA copies are made.
So you can watch the copying happen in real time.
Exactly. The machine measures the fluorescence increase cycle by cycle. The sooner the fluorescence signal crosses a certain threshold, the more target DNA you must have had in your starting sample. It allows for quantification, estimating the relative amount or concentration of the target species DNA. It doesn't usually give you an exact number of individual animals, but it can indicate relative biomass or abundance changes over time or space.
Is that the most sensitive method?
qPCR is very sensitive. There's an even more precise version called droplet digital PCR, or ddPCR. It partitions the sample into thousands of tiny droplets, and PCR happens in each droplet individually. It allows for absolute quantification of DNA copies with very high precision and sensitivity, but it's more complex and expensive.
Okay, so those methods are great for zooming in on one or a few specific targets. But what if you want that broader picture? Like, what are all the different fish species in this coral reef sample?
Now you're talking about community analysis, and the main technique there is called metabarcoding.
Meta-barcoding. Okay, break that down.
The meta implies looking at many things at once, and barcoding refers to using a standardized short DNA sequence, a DNA barcode, to identify species. So meta-barcoding aims to identify many species within a particular taxonomic group, like fish or invertebrates or bacteria, all from the same sample simultaneously.
How does that work with PCR? Do you need primers for every single fish species?
No, that would be impractical. Instead, we use what are often called universal or group-specific primers. These primers are designed to bind to a region of a marker gene that is highly conserved, meaning very similar across all the species in the target group, like all bony fish. But the DNA sequence between the primers has enough variation to distinguish different species.
So one primer pair amplifies that specific barcode region from potentially hundreds of different fish species in the sample.
Exactly. Popular barcode genes for fish include parts of the mitochondrial 12S or 16S ribosomal RNA genes, or the COI gene, cytochrome C oxidase subunit I, which is widely used for invertebrates too. For bacteria and archaea, the 16S rRNA gene is the standard.
Okay, so the PCR creates a big soup mix of barcode fragments from all the different species present. Then what?
Then that complex mixture of amplified DNA fragments, called amplicons, goes on to a next-generation sequencing NGS machine. These are incredibly powerful sequencers that can read millions or even billions of short DNA sequences in parallel, all in a single run.
Millions of sequences. How do you make sense of that data flood?
That's where bioinformatics comes in the interface of biology and computer science. Sophisticated software pipelines are used to process the raw sequence data. This involves quality filtering, grouping identical or very similar sequences together, often called OTUs, operational taxonomic units, or ASVs, amplicon sequence variants, and then comparing these representative sequences against massive curated reference databases.
And databases like GenBank.
Exactly. Databases like NCBI GenBank or Bold Barcode of Life Data System contain barcode sequences from taxonomically identified specimens. By matching our unknown sequences from the sample to sequences in the database, we can assign taxonomic identities hopefully down to the species level.
And the output is basically a list of species found in the sample.
Yes, typically you get a list of identified taxa, species, genera, families, etc., and often the number of sequence reads assigned to each taxon. Now, interpreting those read counts directly as abundance can be tricky because the PCR and sequencing process can have biases. Some species' DNA might amplify more efficiently than others, but it gives you a fantastic overview of the community composition. Using replicates helps build confidence in the detections.
That sounds incredibly powerful for biodiversity assessment. Is there an even broader approach, like sequencing absolutely everything?
There is. That's the whole genome approach, often called shotgun sequencing or, more broadly, metagenomics. Here, the idea is to skip the targeted PCR amplification step altogether.
No primers.
No specific marker gene primers. You essentially attempt to sequence all the DNA fragments present in your purified eDNA extract, or at least a random subset of them. You shear the DNA into fragments, prepare a sequencing library, and sequence it using NGS.
And you compare those random fragments to whole genomes.
Exactly. The analysis involves taking those millions of short sequence reads and comparing them against databases of complete or partial reference genomes from all sorts of organisms, bacteria, archaea, eukaryotes, even viruses.
What are the pros of doing that? It sounds complex.
A major advantage is that you avoid any potential bias introduced by the choice of PCR primers. If a particular fish species doesn't amplify well with your chosen 12S primers in metabarcoding, you might miss it. Shotgun sequencing doesn't have that primer bias. It has the potential to detect a much wider range of organisms simultaneously, including groups you weren't specifically targeting, like viruses or fungi. Plus, you can potentially get information about functional genes present in the community, not just taxonomic identity.
Okay, so broader detection, no primer bias, potentially functional info. What are the downsides? Why not always do shotgun?
Well, there are significant challenges. First, it usually requires much deeper sequencing, meaning generating far more sequence data, which makes it considerably more expensive. This is because the DNA you might be most interested in, say from fish or mammals, might only be a tiny fraction of the total eDNA, which is often overwhelmingly dominated by bacterial DNA.
So you spend a lot sequencing bacteria to find the rare fish sequences.
Pretty much. The analysis is also much more computationally intensive handling, and searching those massive data sets takes serious computing power. And crucially, it relies on the availability of comprehensive reference genome databases. While these are growing rapidly, they are still far less complete than the barcode reference databases used for metabarcoding, especially for many marine eukaryote groups.
So identifying things can be harder if their genome isn't well represented in the database.
Precisely. However, it's a rapidly evolving field. Techniques like DNA enrichment and advances like long-read sequencing technologies, which produce much longer DNA reads, making assembly and identification easier, are helping to overcome some limitations.
So it really sounds like there's a trade-off. qPCR is deep but narrow, focused on specific targets. Metabarcoding is broader within a group, and Shotgun is potentially the broadest, but faces cost, computational, and database hurdles.
That's a very good summary. qPCR for targeted detection and quantification. Metabarcoding for community assessment within a defined group. Shotgun metagenomics for the most comprehensive, unbiased view, including functional potential but with its own set of challenges. The best approach, or combination of approaches, really depends heavily on the specific ecological question you're trying to answer and the resources available. Often combining methods gives the richest insights.
It truly is like having different kinds of lenses to view the intricate web of life in the ocean, each revealing different, fascinating details. Well, thank you so much for guiding us through this amazing world of eDNA today.
My pleasure. It's a field that's constantly advancing and revealing so much about our oceans.
And thanks especially to all of you for joining us today on Saltwater Stories.